continue with our next speaker, who is Dr. Mario de Pinna. It's going to be also a biological talk. He is the second time he comes to our school. Actually, he's finished his PhD in '92, I believe, at City University in New York, under Garrett Nelson.
Garrett Nelson.
Yeah. And from there, he did a tour at the Smithsonian in Washington, then the second tour at Chicago Museum of Natural History, before coming back to São Paulo, where he's a professor at the University of São Paulo, the Museum of Natural History there. He's done several significant contributions to the field of basically, well, like a reconstruction of evolutionary relations. They have some fancy name, phylogenetic systematics, which he's going to define much better for you. And in particular, I think one of the most fundamental works was in the concept of homology in phylogenetics. So, without further delay.
Thank you, Renato. It's a great pleasure being here again. This is always a very interesting crowd, and I love talking to people who are not from my area.
Should we mic him?
Oh, I need the mic?
Yes.
My voice is usually okay.
We want the people in the back to get everything.
Okay, so. That's fine?
Yes.
That's okay? And this?
Just put it here. Anyway.
This is weird. Okay, that's better. So it's always a great pleasure talking to people who are not from my own area. It's really different, you get interesting questions, so it's really nice. Let's see if this works. No, it doesn't. How do I change the slides? Only here in the computer? Or is this?
No.
No? No. So it's in the computer, no problem. I'll stay here. Okay, so Ronaldo yesterday has given you a nice overview of sexual selection and the dynamics related to sexual selection. So today, since the subject is so interesting, I'm going to talk about sex again, but only as a small part of my talk. Just a few examples I couldn't help showing you because they're just too juicy to leave out. So I'll go back to the subject a little bit. My main purpose here is to transmit to you how we express evolutionary relationships, how it is that we show this thing is closer to this one than to this one. How we know that how things are related. That's my main purpose, because this is so important to think straight about evolution. I'll tell you the story later, but actually it took a long time for this to become widely known even within specialists in evolutionary biology after Darwin. So just more on this later. But anyway, it's impossible to talk about evolution without showing as the first slide Charles Darwin and his Origin of Species. All you guys are intellectuals in the academics, and you have seen Ronaldo's talk yesterday, so I won't go into the details of that. But everybody knows the idea of evolution is closely associated with Darwin's life, with Darwin's name. Many people thought about evolution before him, but Darwin was the first one who actually synthesized everything into a coherent theory that convinced everybody that evolution was a fact and proposed a mechanism to explain how it works, which is natural selection. So I found this incredible slide in the internet. I don't know who did it. It's on Google Images, and I got this. This is excellent. The text is not there, it's just the image. But this is natural selection, if you want to know how it works. It's based on 4 very simple principles that can be observed in many natural environments, in many natural systems, in all species. This is exactly what Darwin did. Everything he wrote was based on these 4 basic principles. First, the observation that individuals in natural populations always display variation. They are not clones of each other. They are always a little different from one another. 2, certain individual variations are inheritable, which means that they pass from parents to offspring. 3, certain heritable variations result in differential reproductive success, which means that some of these variations are better in facing certain problems of life than others. And finally, the conclusion of all that, differences in reproductive success due to heritable variation cause changes in the relative representation of such variations in the population. This is a bit more complicated because it's summation of all the others. But basically it means that over a generation, certain variations are better than others at multiplying themselves. And so that's how evolution works. This has been developed over the years into a very coherent theory and completely quantified of population genetics that explains what happens when you have a specific selection pressure, specific variation, and a specific set of genes. This was all developed in the early 20th century and is still being developed today, but it's a quite solid theory. Everybody knows the practical applications and the practical examples of that. This is in every biology textbook. This is the example of Biston betullaria, the moth in England, that before the Industrial Revolution, this form here, which is this one, more visible here, but is also here, mimics the lichens on tree trunks very well. But there is a very rare form, which is black, which is very visible against the same background. So this is strongly counter-selected, because birds can see it. Birds prey on these things. So then came Industrial Revolution, and most tree trunks in England became like this, black. So now this one is the visible one, this one's the invisible one. And guess who became more common? Like the white form became almost non-existent because it was so easy for birds to spot. And then comes the late 20th century and pollution is again not an issue. Trees become like this again. And guess what happens? The white form becomes more common again. So this is a classical example of natural selection caught in the act. But there are many, many, many other examples. It's like, I'm not going to go into that. It's just one that you probably don't know because this is relatively recent. Well, the phenomenon is old, but the understanding of the phenomenon is relatively recent, which is, for example, the codfish. This is just an example. There are similar cases in many of our fisheries resources. Most of our fisheries has restrictions because we don't want to deplete the stocks. Otherwise, we don't have the things to catch anymore. But most of the strategies, all throughout history, have been to limit the size that you can catch, but towards the upper limits. You cannot catch fish below a certain size, right? This is the normal. And this is easy to implement because of the mesh size. You put a mesh size, the small ones go through, the big ones stay. That has been done forever. And people began to realize at one point that codfish, this is the codfish, bacalhau. We're soon going to eat this fish in 1 hour and a half. The bacalhau was getting smaller and smaller as years went by. What was happening? That they're getting so small. People thought, well, it's because they're caught before they have the time to grow to their maximum size. But then someone did a more detailed study and discovered that no. The size of first maturity is getting smaller. It's not just the maximum size. They're getting mature at smaller and smaller sizes. So what's happening? Finally, someone put the pieces of the puzzle together and discovered that what's happening, we're implementing artificial selection on a natural population. We are counter-selecting the big ones. So every big population has runts, things that don't grow. They stay small, and they start reproducing while very small. They have never been very favored by natural selection in natural conditions because they predate the pond. That's why a codfish grows so big so fast. It wants to escape its predators. But then, with our predation, selecting for a small size, the small ones began to become predominant in the population. And that gave rise, actually, to a new branch of fisheries science, which is called evolutionary fisheries. For the first time, incredibly enough, people began to realize that fishing pressures are an evolutionary force. We are changing the genetic composition of natural populations. And that's what makes people more concerned. We're selecting, we're favoring exactly those characteristics that we don't like. Small fish that don't bite, that don't get together, that go to deep waters, that hide in the rocks and things like that. So we're making it harder and harder for us to get what we want. And this is something that only developed in the last 15 years, incredibly enough. And people don't know what to do about it. Because it's very difficult to implement the opposite. Catch only the smaller ones, but not the big ones. We could go on forever on this, on why people think so, but I don't have enough time, too much to talk to you. Natural selection and evolutionary mechanisms in general have been used in many different areas. even in non-biological areas. For example, there is a type of programming called genetic programming or Darwinian programming, which uses principles analogous to natural selection and to evolutionary process in order to to polish a program, and in some cases even to make a program from scratch. This is basically a program that forms an environment where multiple copies of a program are going to compete. And in the end, you have a perfect program and you don't know, nobody knows how it works because it has been selected, it has reproduced by the million inside the big computer, and they have competed with mutation and everything for a certain task. And the successful ones have reproduced more and they win. This is called genetic programming. And if you see the flow charts in their books, I mean, I'm not a programmer, but if you look into their books, it's full of biological names. You see, perform reproduction, copy into new population, perform crossover, this is to increase variation, select two individuals based on fitness, blah, blah, blah. So it's all biological. It's inspired by what they read in evolutionary biology. So Darwin's idea actually works in areas outside of biology very well. Has anyone ever seen this? No? This is an object called a gombok. What is a gombok? A gombok is an object that was predicted to exist in the real world by mathematicians, but what had never been materialized. It is what they call monostatic object, which is a homogeneous object, convex, and that will always go to the same point of equilibrium, no matter where you place it first. It was for the first time materialized by these two mathematicians from Hungary, and it's a very interesting thing. So interesting that I think it is worth showing you a little movie of how it works, because you probably don't understand very well what I'm talking about. This is a gombok, see? You left it in any position, and it goes, okay, it reached equilibrium. That's its equilibrium. Now to go back and forth until it reaches equilibrium. It doesn't matter which position you leave it, it will always go. That's another one. Here it is. Okay, there it goes. And the guys who discovered this, who actually made the object, actually they sell it today for a lot of money. They knew that the thing existed, but they had a very hard time discovering the actual shape that they could mold into an object. It took years. Actually, at one point, they were so desperate, they went to a beach of gravel and started looking at the girl to see if they could have some epiphany of how the shape would be. Eventually, once they got to this, they discovered it was everywhere in nature. That many turtles are like that. Animals that run the risk of staying upside down and dying, they often have gombok-like properties in their back, so it helps them get back on their feet. Many beetles and turtles and things like that. So they made this famous statement in the newspaper, we discovered it with mathematics, but evolution got there first. So the theory of population genetics, which is basically the mathematics associated with natural selection, has been developed for a long time in the 20th century, since the pioneering works by Fisher, C.O. Wright, Haldane, and people like that, all very famous, and these are the names you see in textbook all the time. They're famous equations that I've even seen them printed in biology students so they don't forget. About Hardy-Weinberg equilibrium and things like that. These are the basic equations of population genetics. They describe what happens with the genes in populations under selection or not under selection and so on. And there are many books that synthesize this knowledge. Some have been quite old already, 1966. Of course, everybody knows this guy, Richard Dawkins. He's one of the offspring of this tradition. His semi-scientific book, The Selfish Gene, focused our attention not quantitatively but only qualitatively on certain aspects of evolution that we had not paid attention before. But I want to go back to Darwin, because Ronaldo showed yesterday this book, The Descent of Man and Selection in Relation to Sex, where he explains certain things that in his time were considered as very hardly explainable by natural selection as we normally understand it. And some of them are extraordinarily pedagogical about how selection can go ways that we don't expect, and how it works in conjunction or on the substrate of basic characteristics of a species or the details that differentiate different species. For example, many people ask, how can the peacock's tail be this incredible? It's obviously a handicap for the poor animal. I mean, it's more prone to predation. It takes a lot of energy to do that. It's clumsy. I mean, walking around with that thing all the time, it's difficult walking with these things that live in forests. So it is not the most practical thing. How this thing evolves by natural selection, it looks counterintuitive. Ronaldo explained yesterday how this works. And this slide, again, is a slide that synthesizes the idea very, very neatly. I'll just go through it again because it's such an important idea and it can't hurt to go through it twice really quickly. What happens is that if you have a preference in a female for a trait of a male, or vice versa, but it's usually this way. The male has the trait, the female has the fondness for that trait. Okay, she'll go for this male, the male will go for her. Then you'll have offspring. This offspring will inherit both the tendency to have the trait that the male has and the taste of the female for that trait. Notice, it's a double way. But the taste is only expressed in the female and the trait is expressed in the male. But both inherit both. It's just not expressed in some of them. Like facial hair, for example, in us. I mean, only men have it, but women have the genes that make the same thing. If we give them hormones, they will have. So it's circumstantial, okay? They have the genetic apparatus to do that, it's just not expressed. So it's the same thing. So as generations go by, what happens? They will have offspring that inherit in double dose both the genetic characteristics for the trait and for the fondness for the trait. And what will happen in the next generation? The trait will be more extreme and the taste of the females for that will be even stronger. Over time, that results in things that are completely absurd, like the peacock's tail. When does it stop? Why the peacock's tail doesn't go all the way over the universe? Because at one point, there are other forces that say, no, enough is enough. They begin to die too much. It's like costs too much. So there are other forces, of course. It's not only sexual selection. So everything has a limit. But the results of that, this point of equilibrium, is extraordinary in some species, where you see these extreme cases of sexual dimorphism. And they're not just morphological. Sometimes they're behavioral. Like, for example, this bird that likes to ornate its nest with blue things and builds its nest in a very funny way. It's not a real nest, it's just to attract the female. So it happens in all areas, not just physiology or morphology, it happens also in behavior. Actually, this guy, Geoffrey Miller, he has proposed that this is what guided the evolution of our brain. Our brain evolved as a secondary sexual characteristic driven by sexual selection. And then you ask, well, why is it that not only males have big brains and females have mouse-sized brains? Because that's what happens in many other traits. Because in cognitive abilities, you need the other one to understand what you're doing. I mean, you can do very elaborate things, but if the female is not able to recognize that, it's useless. So the two brains evolved at about the same size, so one could do things, the other responds, and then the first one responds to the second, and there's this interaction. That's his theory. It's probably right in part. And we find examples of this everywhere when you look at animals. It's very normal to have a strong sexual dimorphism. Since I'm a zoologist, I can't help showing you a few interesting cases. Like this is in mammals, the largest case of sexual dimorphism in size. This is the elephant seal. The male is about 10 times the size of the female. But there are other cases which are the opposite. For example, these deep water fish, the male gets so small that it becomes a parasite on the female. At one point, this is a better picture, it's a freshly caught one. This is extremely rare to see. These fish usually are completely damaged because they come from too deep and they're too fragile. This is perfect. This is a female, and these are two males attached to it. There can be more than one. So what happens is that in the beginning they're all the same. Eventually the female grows and the males find the female, attaches to it and never lets go. It's a marriage for life. The male eventually loses eyes, loses mouth, loses everything, and basically becomes a little sperm producing machine attached to the body of the female. It does nothing. They have a hormonal integration. The blood vessels of the female go inside the male to feed it. Quite a fate, huh? And then, since there's hormonal integration, when the female releases the eggs, the little male has an orgasm and releases the sperm. He doesn't even know what's happening anymore. It's just, he became just a little organ. And the organism is bisexual for all practical effects. Why is that? Well, these things live in bathypelagic region. It's like one fish for every 3 kilometers of cubic water, and they hardly move. So if they meet a partner in their lifetime, they're lucky. It's never to let go, otherwise they'll never see it again. But this is not just a thing of big animals. This is schistosoma, for example, and this is the male, and the female is in here. The female is this thin worm in this cleft, and when the male finds a female, they fall in love immediately, and the female gets close and is immediately embraced by these ridges and never leaves. So this is the male, this is the female. So it happens at all levels, not just big animals. It happens everywhere. And sometimes they get so different that selective pressures in the environment are different, because it is different being small and being big, for example, being bright and being of a dull coloration. The selective pressures, predation pressures, they're all different. For example, this is the largest case of sexual dimorphism in size in cichlids, which is a family of fish. This is the male, this little thing is the female. Notice how different they're in terms of color, because the female has to hide well, otherwise someone's going to eat her. Because she's too small, even the male could eat her. These are two adults. The male is past the size of predation, so he can be showy, no problems. And he has the normal competition with other males for baseball, mostly on brute force. That's why they get so big. But why doesn't the female grow big too? Well, that's when the details about the life history of these things come into play. These fish, they spawn inside these shells. You see many of them here. That's where the female puts her eggs. These shells are everywhere. If they don't do that, if the eggs are outside, they're immediately eaten, so they don't survive. So the female has to put the eggs there. In order to do that, she needs to be small, because the size of the shell doesn't change. Being so small, she has to have a different coloration. So eventually they get very different, even though they're the same species. This is what's called disruptive selection. It's selection that tends to make things go apart. The name's exactly what it means, disruptive. Okay. Of course, there are many examples in invertebrates. So this is the opposite in terms of size. The female is many times bigger than the male. This is one, well, it's not the same species, but you see this spider is very similar in gardens everywhere, including here. This is the male, this is the female, these are two adults. It's an extreme case of size difference. Of course, if you look into the details of the lives of these spiders, the males eat very different things than the females. They build different webs. Of course, they're different animals, so they're subject to different pressures. Not all invertebrates, most invertebrates is the female who is bigger, but in some of them it's the opposite. The male is bigger. For example, in this one. This is one isopod, which is a freshwater, in this case, freshwater crustacean, and the female is here in the belly of the male. The male actually has a specialized pair of legs. You see there's one missing here. This pair of legs is holding the female, and she stays there the rest of her life. And what happens if she leaves for 1 minute? Another male comes and tries to grab her from the first male. And they start fighting for her, and her legs are pulled apart and she dies. So she has to be small to be under the body of the male all the time, so she's not destroyed. So there is selection against getting even a little bit bigger. Ronaldo yesterday showed us this, the case of the parthenogenetic lizards. And we have one, I don't know if it's the same one you showed yesterday.
Same genus.
Same genus, yeah. This one is parthenogenetic, and is a hybrid species between these two other species. I don't know their names, but you later can tell us. It's a hybrid species that became parthenogenetic, which means that the female reproduces without males. That's a very interesting case, and occurs in many groups of animals, but always in very isolated cases, small groups, short-lived. That's why we discussed yesterday about the validity of sex. I won't go into that because we already discussed it yesterday, but this one is, for for my favorite group, which is fishes, is worth mentioning. This is what's called a sexual parasite. This is a female of Pisidia formosa, and people thought it was parthenogenetic initially. But then they saw the female copulating with males of another species that had males. And they said, oh, eventually they make biparental reproduction. But then they studied it genetically and discovered that there is no genetic input from the males. She's copulating with males from other species, not her own species. Her own species only has females. What happens is that the sperm of the male acts as a trigger for the development of the egg. But the genetic material is not incorporated. So it just spits out the genetic material when the time comes to form the zygote. So it's basically parthenogenetic but needs males. And since it doesn't have males, it parasitizes males from other species. And it got specialized in being particularly attractive to males of other species. So it's a big price if you have these things around for one of these fish, because the females are more attractive than the respective females of their own species. So the males all want to copulate with them, leaving no offspring at all. So this is why they're called sexual parasites. It's not a porn movie, it's it's a biological term. More recently, people have discovered really odd things that people thought were impossible, which is the opposite. Species that only have males, they don't have females. They're very difficult to detect. Why? Because in order to create a new animal or plant, you need cytoplasm. You can't have just the sperm. You need mitochondria, you need the cytoplasm. So you only detect that genetically. And it's only recent that such methods have been available. But for example, this tree is one tree that only has males. This is a dioic tree. It's the Sahara cypress. It's very rare these days, but the species is very resistant to drought. It can tolerate very dry conditions. It exists in the middle of the Sahara Desert, a tree this size. And how does it reproduce? Only males. Now they understand what it does. It uses the female trees of other species, and it inseminates them with the pollen. And then, once the zygote is formed, the genetic material of the female is completely excluded, and the seeds are exclusively male and exclusively of this species. Why they're doing that? It's weird. They're a species that is much more resistant to drought than either of the closely related species. That's why they're going extinct, because now they have no more trees to parasitize sexually, because the only trees are where they live. So, but they have spread all over the world. And there are all kinds of odd things, like this. For example, these are two snails in the process of copulating. What many people don't know is that we are watching gay sex here. Because gay sex in many snails, not all of them, pulmonate snails, some pulmonate snails, but a big number of them, is mandatory. Otherwise, that's part of how they reproduce. Because they are hermaphroditic. But they're not hermaphroditic simultaneously. They're first male, and then they become females. So when two snails meet, they're both male. They both produce sperm. So they exchange sperms, so they do sex. The one gets the sperm of the other. Then they separate, each goes his own way. And then they turn into females, using the sperm of the other male to fertilize his own eggs, not her own eggs. So, yes, there are all kinds of, if you want to really see all the variations imaginable in nature when it comes to sex and other processes, go to natural history. This is also something that many people find unbelievable. We always think about sex as being two things, male, female, male, female. We even see science fiction movies and books that sometimes say, oh, there are 3 sexes in this planet, blah, blah. We have this thing on Earth, on our own planet. There are many animals that have more than 2 sexes. They're usually very small and unknown. But, for example, ciliate protozoans, they can have up to 100 different sexes, which is a very good strategy, because if you have a 1-to-1 sex ratio, randomly, you have a 50% chance of finding someone you can make sex with. Well, I guess the chance of making sex is 100% if you really want, but to leave offspring is 50%. But if you have 100 different sexes, you have a 99% chance of anyone in the population being able to do sex and leave offspring with you, right? Because everybody's different. You just have to avoid 1 in 100 is exactly the same sexual composition as you are. So this is also very weird and many people don't know. We talked yesterday about rotifers. which is the only major group which is highly diverse, old, and completely asexual. You don't have males in this group, only females. And worse still, worse still, it is possible to construct their evolutionary relationships, which should be impossible because they are clones. So they shouldn't have a history of descendants in the usual sense that allows the usual methods to reconstruct phylogeny. So what's happening? The thing was solved a few years ago. They discovered that these rotifers actually do a lot of sex, but they do sex not among themselves necessarily, and not with contact, but they do sex with DNA from the environment. They take bits and pieces of DNA floating around in the water and incorporate in their own genome. So they do a lot of sex. That's why they're so diverse, and that's why they have been surviving for so long. That's the paper that discovered massive horizontal gene transfer in delicate rotifers. Basically, sex is this, is exchange of genetic material between different individuals. Anyway, an extension that took a long time to happen of natural selection to explain some phenomena, again, that were apparently counterintuitive for selection, is the development of social behavior and altruistic behavior. Like the sentinels that warn other members of the group that there is a predator. Usually this guy is more heavily predated upon than the others because it's yelling while the predator is coming, so the predator is going to see it first. So what possible force of selection would drive this kind of behavior to evolve? This took a long time to be discovered. It applies also to social insects, applies to warning coloration in many animals, because if your predator hasn't learned yet, you increase your chance of being eaten, right? It will eat you first and then discover it tastes really bad, but you have to die first for you to learn. So all this was explained, to my view, very well by this guy, William D. Hamilton, in the mid-60s. Some people say this guy Hamilton is the most important evolutionary biologist since Darwin. I kind of agree with him. He was a genius. He was absolutely brilliant, and he explained lots of things. For you young people who are starting your careers, don't get discouraged when your paper is rejected by a major journal. These two papers, 1 and 2, were rejected by 17 journals before he could get them accepted. And they're considered two of the most important papers in evolutionary biology in the 20th century. So yes, it's a word of hope for us. Because they're so different. And Hamilton also explained many, many other things. This guy, for example, is Peter Medawar. Incidentally, this is the only Nobel Prize who was born in Brazil, ever. He was born in Petrópolis. Yeah. Of a, Lebanese father and British mother. And he went back to England when he was 8. But hey, he was born here. He's listed in Wikipedia as a Brazilian Nobel Prize. So he proposed an explanation for senescence, why we get old, why we have these characteristics that we perceive as getting old, somatic degeneration and things like that. His idea was very good, but the one who made the real hard work, you see parts of the pages here, was Hamilton, showing how this was a correct theory. That natural selection naturally will tend to push the deleterious effects of genes towards the later parts of development. So the same gene that makes you strong and healthy at 20 may be the same gene that's going to make you have a brain cancer at 50. It doesn't matter, because at 50 you already reproduced. So the deleterious effects are pushed towards the later phases of development, and that explains senescence everywhere. It's a bright explanation, and I think it's completely correct. Hamilton, of course, is most famous for this little thing, which is basically the inequality that says when kin selection is going to happen. Basically, it's when the coefficient of relatedness will be bigger than the ratio between the cost of cooperation and the benefit of cooperation, whichever way you decide to measure this. This is the basis of the explanation for altruistic behavior, because if you see most animals that show altruistic behavior, they form groups that are related by kinship. So you can quantify this. For example, you're related to yourself completely, but you're only related 50% your parents or brother or sister or your child. But as you go further from yourself, the relatedness gets smaller and smaller. So once people say, he said, yeah, I'm willing to die for 2 children. But to die for a cousin, you better put 8 cousins there. I couldn't die for a cousin. I don't have 8 cousins. So, and then when you contrast these numbers with nature, this is exactly what you find. It's very interesting exactly because of this. The equations, actually, they make predictions about the world, and you go there, check them, and they're there. It's exactly the same number. And in some cases, I'll get to this really quickly, but in some cases, it's even, let's say, a little politically incorrect to get these data. Because for many years, there are no data about infanticide of adopted children versus real children. They thought this posed a wall of prejudice against adopted children. So these data are not available. And finally they got these data, collected the data newly, because it was not available in social files of most countries. When they got the data, it was exactly what social biology predicted, what kin selection predicted. Adopted children are killed by their parents at a much higher rate than their natural children. So that was predicted, and the data was specifically sought after to prove that. Eventually, this whole thing became what's known as social biology. I don't know if you guys heard about this, but this is, how can I say, practically a curse in the social science. Social science sociologists and anthropologists, but they hate this kind of thing. They think this is the end of the world. No, 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 Nazi stuff, it's impossible. But the theory is absolutely brilliant. It explains how social structures are originated by natural selection and maintained over time, including in men. We are a social animal, just like, not social, not really social, but whatever you have of social is explained by the same principles that guides and controls the evolution of social behavior in many other animals. It became very, very famous. Leo Wilson is a celebrity. At one point he couldn't go out in the street because people would throw eggs at him. But well, now the distance is making people a little less vitriolic about it. Obviously the association they did was sociobiology and eugenics, the movement of the early 20th century, the first few decades of the 20th century that tried to improve the human race by selection. Only some people can reproduce, the others cannot reproduce, and things like that, which is something incredible. If you look in Google, look for old eugenics ads, you find things that today are very shocking. For example, the most merciful thing that a large family does to one of its infant members, large family, poor. You can read poor, is to kill it. Okay, easy solution. I mean, this was actually being said. People said these things. So eugenics is not a movement that's very endeared to people today. So in making the association between social biology and eugenics, you basically put social biology under a very bad light in society, which is a very low blow to give social biology. Didn't deserve this. It's a brilliant theory. Okay, but this is just part of what I want to say. I want to go now into another aspect of my talk, which is something that I mentioned at the beginning that I wanted to insist on you as non-biologists to understand, because I think it's very worth. It's something that will help you understand evolution in a much clearer way, and let's say much more rigorous way. The only illustration in the origin of species, there's only one illustration, is this one, is a phylogenetic tree. Why Darwin put a phylogenetic tree there? Because he thought it was so wonderfully pedagogical about the process and the dynamics of evolutionary diversification. He explains what happens here. This is time, and this is the history of diversification of this lineage, and that lineage, well, this is not standard today, but you understand more or less what this means. This is very, very different from what people understood as evolution before Darwin. There are many people talking about evolution before Darwin, including Lamarck, the most famous one. Lamarck had a theory of evolution which was very different. For Lamarck, you had spontaneous generation all the time. And as soon as a new living form pops out on the ground, it begins evolving. But it evolves in an independent manner, following a predetermined track. So it starts as some microbe, and then starts evolving, and over millions of years it will turn into a mammal or something like that. So since you have spontaneous generation all the time, that explains why you have the diversity of living things on this planet. Because otherwise you would only see this, right? No, but since it's appearing every time, there are things that just popped out today, and things that popped out billions of years ago. So they have lots of time to evolve. So there is no relatedness. It is almost like a law of physics, Lamarckian evolution. There is not the branching process that we see that Darwin discovered. It is a different thing. Many people still think like this. Even nowadays, people say, "Oh, how can evolution explain the evolution of the human brain? It's such a wonderful machine, blah 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 blah, blah so enormous." Well, you don't need to explain the evolution of the human brain. 99% of our brain was already inherited from mammals, from primates, blah blah blah. It was already pretty big. We just did a little more. So that's how evolution works. It builds upon what's already there. You don't need to explain the evolution of things from scratch. You only need to explain that little bit that separates that species from its closest relative. Right? So there's no great enigma. Most of what we are, was not evolved as a species in Homo sapiens. It evolved a long time ago. Part of it from the beginning of life. We only need to explain a little bit. All the rest was inheritance. Very easy. Incredibly enough, the language to express evolutionary relationships took 100 years to be developed after Darwin. For 100 years, people were messed up in their heads about how to express evolutionary relationships. You saw diagrams like this. This is post-Darwin. I mean, what is this? I mean, how do you understand this mess? There are names here, there are names here. It's obviously something really strange. But well, this is from Haeckel, okay, 19th century. But if you go into the 20th century, you still see things like this, which are very difficult to interpret. How you delimit this group? I mean, how are these lines drawn? Everything's kind of subjective, very difficult to. There's a large degree of subjectivity in these diagrams. And you see ad hoc concepts like to say we are very different from other primates. We have a metaphysical concept called an adaptive zone, which is not we are very different from the others. This kind of thing. This is all messed up. It was only with that book that I showed you in 1966 that things changed into the marvelous clarity of what we call cladograms, which are modern diagrams to express phylogenetic relationships. Notice that these diagrams don't have species as ancestors. All relationships are just hypotheses of relatedness. And they usually have this kind of structure. They can be drawn many different ways, but the basic information is, sorry, some of these slides are in Portuguese, because I just picked them from different talks. Okay, so what is a terminal? A terminal is an actual entity from which you get data, an organism. All the rest below it is hypothesis of relationship. So it doesn't really, it's not based on something you observe. It is based on something you hypothesized, based on the data you see at the top, by various methods of phylogenetic reconstruction. And all this means is that these three elements here are more related among themselves than any of them is to an element here. That's all it says. It says nothing about actual ancestors, about actual ancestor-descendant relationships. It just says a statement about proximity of descent, propinquity of descent. And that's all the diagrams today say. But in being so simple and so tight, they're much more powerful than the fancy trees of yesterday. For example, they allow us to understand what a group of animals is. What is a mammal? I mean, most people don't think about that too much. What is a mammal? What is an insect? Why do we recognize a group such as mammal or insect? The reason is these groups have a historical coherence. They all descended from the same ancestor, and only this ancestor. So a group is actually something that includes an ancestor and all its descendants. That's what a group is. That's why we recognize things like birds, like mammals, like crocodilians and things like that. That's why groups exist, because they had a single species which was the ancestor of all of them, and then they diversified. So phylogenetic analysis is this branch of biology, of evolutionary biology, that devotes itself to untangling these things and developing methods for constructing phylogenetic relationships. Just still here. For example, you see this group in yellow here, the big one. This is a monophyletic group. It's a real group because it includes an ancestor and all its descendants. But if you get a group formed by Mammalia and aves, for example, which could be like warm-blooded vertebrates, this is not a natural group. They don't share a common ancestor. Or more precisely, they do share common ancestors, of course, but it's not exclusive to them. It's also the ancestor of many other things which don't have warm blood, for example. And another common phenomenon is what's shown here in blue. It's an incomplete group. This is also not a natural group, which is the group of reptiles. Reptiles, in order to be a natural group, have to include aves too, because then it will be a natural group. An ancestor and all its descendants. If you take away one of them, it's no longer a natural group. So groups in classification are real things that existed in the past. And phylogenetic analysis discovers that. Yeah?
So that's the current trend is that Mammalia has no connection with Reptilia. That's
No, it has. But Reptilia is not monophyletic. Reptilia has many different branches and they do not have an exclusive ancestor. They're not all shown here because there are many fossils. You have fossil groups here, 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 everywhere. If you, what we have, the trees we have today of living things are just a shadow of actual diversity in the past. Because 99.99% of all species that existed are already extinct. Yeah.
okay.
And it's always been like that. It's no major disaster. Disaster is what we're causing extinction now, environmental disaster. But over history, in geological time, species go extinct regularly. It's normal. So if you have fossils, you fill in this tree. Yeah? There are many things that exist in the past that we don't see today. So the relationship between these diagrams and the hierarchical classification are obvious. So all classification today is based on these diagrams. I won't go into the details, of course, but what is really interesting is that these diagrams also allow us to map the evolution of specific traits. We can look at these trees and understand how the evolution of specific characteristics of the organisms here evolved. For example, it's a very didactic diagram, but you can say 4 legs evolved here. This is possible to say in this kind of diagram. You can plot the change that you see. And this is what allows us, for example, to say that flight, for example, evolved 3 times in vertebrates. Why we know that? Because the wings are different? That's not enough. Many things can be very different and be the same. We know that because when you make a tree of vertebrates, this comes in one branch, this second one comes very distant from the first one, from bats, and finally birds is still very far away. So we know there's 3 kinds of wings, only have evolved independently, so they're convergent. That's why I say they evolved 3 times. You need a phylogenetic hypothesis or a diagram to be able to say that. And that allows us to say a lot of interesting things. For example, stick insects have lost wings many times. And a detailed phylogenetic analysis allows us to show that wings were lost and then regained again. We can only say that because we have this kind of hypothesis there in much detail of all the species and how things change in each branch. Otherwise, you can't say that. If you don't have a hypothesis of relationship, you can't say anything about how things evolved. And they're very useful. It's a pity that Ronaldo is not here now, but because this is, covers this, he just stepped out, his area of specialty. For example, you're bitten by a snake, and not all snakes have specific antivenoms for them, right? You have antivenoms of different kinds that cover hundreds of species. So, for example, if you're bitten by a king brown, there's no specific antivenom for king brown. Which one are you going to use? Well, you go to a cladogram of these snakes. You see the king brown is here. What are its closest relatives? These ones in green. So you get an antivenom of one of these snakes. You shouldn't get an antivenom here. It's too far away. You increase the probability of surviving if you take something from a snake, made for a snake, which is phylogenetically close to it. So you should get one of the one of these species here that are shown in green, in this green clade. So it is useful. Yeah, it can save your life. And you ask, well, how can you be sure about what you're saying in relationships when these things happened so long ago and nobody observed them? There are many methods that are used for that, mostly statistical and based on congruence. But there is also a small branch of what they call experimental phylogenetics. People who made artificial phylogeny of bacteriophages, which are viruses that eat bacteria, and they made their own phylogeny. They separated the ancestral lineages, and they let them diversify, blah, blah. They evolved fast enough so you can do that in the lab. And eventually they tested back all the methods to reconstruct the phylogeny and confirmed that they actually are able to reconstruct the phylogeny that was artificially made. So yeah, there are many independent lines of evidence that show that we are on the right track. Of course, today we have a phylogenetic hypothesis for the whole of life, or the whole of life that we still have living things there. More precisely, probably the base of the tree was more something like this, because there is so much horizontal transfer. But this doesn't mean you cannot make phylogeny. Phylogenies are normal in this case too. I don't know if any of you have heard about LUCA. LUCA is an abbreviation for Last Universal Common Ancestor, which is the last thing that was the ancestor of both archaea, bacteria, and eukaryotes. Archaea are one kind of bacteria. It's very different. It looks like a bacteria, it's a prokaryote, but it's very different. It's as different as eukaryotes are from bacteria. So these are the three main domains of life, and one of them was actually a fusion or reticulation between the other two. Recently there was this paper where they tried to, they did something weird. They got all the genes in gene bank for prokaryotes and a few eukaryotes, and mapped all these genes for protein coding, took the ones that were not protein coding, genes that code for protein, and made several filters for where they occur in this universe of prokaryotes and eukaryotes, and arrived at a set of 355 genes that they think have to be the genes that were in the last common ancestor, the LUCA thing, last common ancestor of everybody. And these genes, they say we're very surprised to know that, but indicate something that lives in one of those thermal vents under the ocean. They have protein synthesis for high temperatures, for presence of sulfur, this kind of metabolism that indicates it was something that lived. And then they say, so life originated in something similar to thermal vents. We'll see. This is too recent to be sure about. They did some dirty tricks, like many of the genes that are related to oxygen metabolism were there, but they filtered. And that doesn't make sense. There is not oxygen there at this time too. So that was criticized already. So we'll see, but people are trying to do this kind of thing. It's all through, of course, phylogenetic analysis. It goes into all that. Without this kind of method, you can't do anything. You can't know anything. Of course, if you want to go to the origin of life, what you have to, that's what I think, you have to look at the ribosome. The ribosome is the key to the origin of life, I think. And many other people think too, not everybody. Especially this particular P site, so it's called the P site. This is probably the most ancient part of the molecular machinery in all living beings. It is the only thing that is exactly the same in everybody, from retroviruses to mammals, to insects, to deep water fish, everything. This little portion of the ribosome that does part of the link between the peptides that will form protein. It is an extremely complex machine, but this little part is very small. And this is probably the oldest part still in the bodies of living things. Here's an animation I found for you to see how this works. There are better animations, but they're not public. Yeah.
In that case, is the mitochondrial RNA or?
No, sorry, it's RNA. It's ribosomal.
Right.
Did I say mitochondrial?
Yeah, but.
No, sorry, it's ribosomal. Yeah.
Okay, but in the picture, in the previous picture is mRNA, but it's just a.
Oh no, it's messenger.
Oh, it's the messenger.
Yeah, because the ribosomal RNA, the RNA has three forms.
Okay.
The ribosomal one, the messenger, and the transfer. So that's, no, this is all RNA. Yeah?
You want to finish?
Sorry?
Now, I was going to pick you on the tree of life quickly. Because if you say that ribosome is the commonest part of all the living things you find, you probably would have to reshape the tree of life to include something that's shared life characteristics, but it's not eukarya or archaea or bacteria.
Oh, certainly there are
Is that
many
correct?
things. Yeah, absolutely, absolutely. I think what they call LUCA is very recent, actually. I mean, compared to the origin of life,
Okay.
it's probably hundreds of millions of years in the front. Because the genes they got were genes that work, genes that depend on proteins. The thing that had all these things was already a functioning organism. They tried to attract the limelight by saying, oh, this LUCA that we discovered was half life, half non-life. And people ask, why? Because they needed external elements for their metabolism. That means I'm also half living because I go to the supermarket to get my food, right? That doesn't mean anything. Every living being requires things for we need vitamin C that we don't produce. We need many things we don't produce. Not just for energy, but actually for our metabolism. And as far as I know, we're quite alive. Huh? Yeah, so far. So this is an incredible machine, and people are paying a lot of attention to that particular spot. And there have been even studies about how the ribosomal RNA actually was built around this nucleus. This is the very functional nucleus that makes the binding between peptides that will eventually form the protein. But, of course, as you proceed, the ribosome gets bigger and bigger with other things. It's a very complex structure. Interesting, RNA is a nucleic acid, right? But it's also an enzyme. That's why we call them ribozymes. So they are kind of ambiguous in what they do. They both accumulate information and they act as an enzyme. That's why people were attracted to RNA as the origin of life for the first time. Much of the phylogenetic research today is, of course, based on our wonder molecule DNA, because DNA is so marvelously informative because it's so big. It has billions and billions of bases that go on forever. And if you know the sequence of these bases, you can compare different organisms and infer how they are related. And this is the subject of phylogenetic analysis. For example, if you have 4 organisms, and one specific spot of one specific gene, they have A, and the other one has G and G, you can conclude that A was here at the base of A and B, and G was at the base of G and G, of C and D, sorry. Because it's just the simplest explanation. It changed from A to G somewhere along this line. But of course that doesn't need necessarily to be so, because the probabilities of these changes may change things. So any of the other hypotheses may be also correct depending on the parameters you're using and specific parameters for the evolution of those genes. So we have models, statistical models that are more or less complex depending on the case, that change these probabilities, sometimes asymmetrically, in various ways. I'm not going into the details, you'll be bored to death here if I go into all the little details of how probabilistic phylogenetic analysis is done, but it's a very active area of phylogenetic research. Of course, at the base of everything is the comparison of sequences of letters. But there are many, many details that go after this, and methods have very different performances in finding the right tree. And there are people who devote their entire lives to doing this kind of thing, simulations in comparison with actual things. Let me see where I am in my time. I still have some time, because we started late, right? Okay, so one thing that many people don't realize, they think, oh, it's very easy to make a phylogenetic analysis based on DNA. You compare how different they are. So if you find things that are very close, like us and the chimpanzee, you find only 2%, 2.5% of difference in most genes. What's this difference means? It means that you have A, C, 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 T, A, T, 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 T. The chimp will be almost exactly the same thing, except for one T instead of an A here, one C instead of a G here. No. So you can quantify these differences and see the known hits, quantify them, and say, in a table, this is closer to that. It would be very simple if it was like this. Unfortunately, DNA evolution is not like that, for a very simple mathematical reason. You get 2 identical sequences, and you start mutating them randomly. In the beginning, they will get different at the same rates of mutation. One mutation, one difference that you can detect. And the curve goes like this, almost linear. It looks linear. As time goes by, what will happen? You see that you change it to A to T here. And then eventually there'll be another mutation in this T. And some of them will change it to A. So the difference will decrease when you compare. And in some cases you see AT, okay, one change. But actually this T here was a result of a first change to C, then another to G, then another to T, then another to A, then another to T again. So you're underestimating brutally the amount of divergence in sequences if you only look at the differences. So you have to correct that. Actually, the curve of difference does not go like this. It goes like this. At some point, they cannot get any more different, no matter how far you are. For example, ribosomal sequences, they're very, very conservative. So you use the ribosomal sequence comparisons to study the really deep divergences of life, things of billions of years old. If you're going to make a phylogenetic analysis based on ribosomal DNA among mammals, you won't find anything. They're all the same. There are very, very few differences. You need to go really deep. So you have to choose your genes and calibrate all that and then correct according to this curve that I mentioned. Okay, but this is all technical stuff. We can talk a little bit about ourselves. Of course, this has been one point of attention since people began to compare animals in general. Are we part of this? Are we part or not part of this? So this is just one phylogeny that includes our genus Homo, and then Pongo, Halobates, Macaca, and all the other, a few of the other primates there. What is interesting here is that with this kind of divergence in nucleotide sequences, you can also estimate time, because these mutations, in some cases, they accumulate at a quasi-fixed rate. Not completely, they can change in branches, they can change everywhere. This all has to be calibrated. But if you make all these allowances, you can tell the date of divergence of certain things by the number of nucleotide changes you have in the comparison of their DNA. So you arrive at not only a phylogenetic hypothesis, but some estimates of the time of divergence. But this kind of divergence has to be calibrated. How do you do that? You can do that usually with fossils. You get fossils that you know where they go in the tree, and then based on that and the age of that fossil, you have a minimum age for that kind of divergence. And you can then use these numbers to calibrate the entire tree. You say, but can this change? Can these rates change? Yes, they can change. Yes, you should be careful. Don't go too far. You have to stay more or less in the neighborhood of where you are for these calibrations to be reliable. Otherwise, you begin to get distorted results. The rate of heterogeneity of a tree is actually the biggest problem in any kind of phylogenetic analysis. Like here, you can have, let's say, one change every million years, and here you can have one change every 10 million years. That distorts the tree in the analysis completely, so you have to correct for that. Of course, there are other ways of testing this. If you have access to some biogeography and can date the events and can correlate these events with specific points in your tree, then you have an independent way of evaluating these calibrations and see if everything is right. Of course, this is all very difficult, takes a lot of work, but it can be done, and it is done every once in a while. And once you have things that are well calibrated, you can do really cool stuff, which is, for example, dating the Hawaiian Islands according to their divergence from a specific group of organisms that only lives there or diversified there, because their history of diversification is going to be the history of the islands, of how they originated by volcanic activity. So you basically can use biology to reconstruct the geological history of an area. This is a whole area of study called biogeography today. Now, we're talking about how you understand trees, how you understand diversification, but how in health species split, why they separate. That was the problem that initially triggered Darwin's interest in the subject. You want to know how species multiply. Why do you have many species instead of just one? Well, the reasons for that are historical and contingential, if I can pronounce it this way, for the history of each case. Why? Because what keeps a species together? What is it? Why we are the same species? Because we are exchanging genes all the time. Sexual reproduction assures that we change genes with each other. Therefore, we never get too different from each other, because genes are always mixing. This is what glues species together. Species stay more or less uniform because of this. But place a barrier between part of the species and the rest, and differences begin to accumulate. Because what happens on one side will no longer travel to the other side. It will stay there, and over time you have divergence, and they get more and more different. And this is what drives speciation, which is the process of lineage splitting. It can be caused by a geographical barrier, for example, continental drift, a river that appears in the middle of a forest. That depends on the animal or the plant. Some barriers are very effective for turtles, but they'll be not effective for a bird. So this all changes. And over time, you have this process, this mosaic of splitting and mixing back again that results in things like the Amazon forests, with millions of species living on the same spot. But in essence, the process of speciation is very, very simple. It basically is based on a barrier that keeps genes from flowing freely throughout the population. If there are some cataclysmic event that kept us in this room for 10 million years, isolated from the rest of the world, and we started doing sex among ourselves and procreating, in 10 million years, this might be a very scary perspective, but in 10 million years, we would be a different species, or much before that. Why? Because we would be here, isolated, a very small population size, under different conditions. Anything that happens in our gene pool would not go outside, and vice versa. So it would begin to diverge, and eventually become a different species. That's how speciation occurs. It's always like this. Sometimes distance alone is a sufficient factor. There are simulations now that have been matched in actual natural populations that show that when you have a very large population, parts of it, the probabilities of gene exchange are very different from the center to the periphery. So it is possible to have speciation without a barrier if you have distances. Distances, of course, relative distances for the specific creature that you're dealing with. This is unnecessary. So every lineage splits like this. You have an ancestral species, then there is some barrier that splits it in two. From this point on, you begin to have differences that accumulate in one side and cannot go to the other side and vice versa. Of course, when you have polymorphism here, this polymorphism is going to be inherited for some time in the two. But they may or may not be maintained in the long term. So here, for example, this diagram shows that there's the blue and the brown versions of this thing. Eventually, the blue one becomes absolute here, and the brown one here. But this is a stochastic process. Maybe related to selection, maybe not.
Some species, as I understand you speaking, they co-evolve. They exist in parallel. They work together. Let's say the human species going back. There was kind of this living together, Homo erectus and Neanderthal, and so I don't know the timeline.
Sapiens for a while.
What causes, except for the environmental pressure, what causes that one species stays and the other one dies if they are so similar and they live in the very similar environment? What causes that?
Well, usually it is something related to their competition. If you have two species competing for the same resource, usually one of them goes extinct over time. Not immediately, they don't need to eat each other or anything like that. They're just competing, but generation after generation, one of them gets smaller and smaller population sizes until it disappears. So it may be that. In our case, because we are war-making species, we probably killed them all as soon as we met them. It's not unusual. It happens too for species that have territories, that compete. This is not unusual. Although apparently with Neanderthalensis, people don't know exactly, because many populations of Neanderthalensis actually died off before they met Homo sapiens. So, I don't know how true that is in all cases. Things can go extinct without competition, without anything going against them, just because bad luck. It happens. There is a lot of contingency in these specific histories of specific taxa. I mean, dinosaurs, big group, fantastic evolution, finally refined for different environments and different habits and everything. So the meteor comes, boom, okay, everybody dies. Nobody cares. I mean, it's not their fault or their merit to survive. Well, one group survived, birds. They're still wrong, but it's very little compared to what was there. Right? So yes, sometimes you can explain it by very specific factors. Temperature change, climate change in general, change of resources, blah, blah, blah. Sometimes you simply cannot. It's very difficult to spot. What you can say is that it happened, when it happened, and what's the structure of diversification that led to the extinction of some lineages. These things you can actually test directly. The others, you need to go specifically case by case, because these causal factors are very multifaceted. Okay, so this is just a little explanation of what people mean by coalescence, which is an interesting concept that's everywhere. When you say, oh, Lucy was the most recent coalescence of all the humanity, well, what they did was the calculations to see mitochondrial DNA, which is inherited exclusively matrilineally by the mother. Mitochondria only exist in the cytoplasm, so it's exclusively a mother inheritance. They made the calculations and discovered that there was this ancestor there some time ago. And they say, oh, Lucy existed. They called her Lucy. Well, many people think Lucy was like the only specimen of that species, of our species, that was the ancestor of everybody. No, she was just the bearer of the version of mitochondrial DNA that everybody inherits today. But if you went to the population where she lived, it was a normal population, with lots of males and females procreating all the time. Normally, she might not be different from anybody. It might be a purely stochastic process that eventually resulted in her being the mitochondrial ancestor of everybody. So there are a lot of misconceptions about how these things actually work. For example, one thing that cladograms helps us, I didn't mention this before, but it's very important, is that when you have a lineage split, okay, one species divided into two, which species is older than the other? None. They're the same age, because they both originate in the same point in space, in time, sorry. So they're the same age. If this was 1 million, this is 1 million. By definition, there's no other way, there's no way out of this. And that applies to all the rest of the branching diagram, which means the following thing. For all things living alive today, all of them have the same age of evolution. We have as much time of evolution as the bacteria in our gut. The same, because we came from the same point. And since time is ultrametric in a tree, it's always the same time, it's all the same age. So lineages have all the same age. It's the splits that occur at different ages, okay? So this is a very important thing to actually melt away some of the misconceptions about how evolution works. There are one species is not older than the other. You can say it changes more or less relative to another one. But the time is the same, all right? Okay, so there are many models of speciation that explains what can happen. They get different names. There are actually many more names than this. So allopatric, which is the classical case, peripatric, which is peripheral isolation, parapatric, which is a variant of peripatric speciation, sympatric speciation, which is on the same spot. So all this happens in biological systems anywhere. And there are many different mechanisms that explains each of them. We can go into that if you guys are interested later, but I'm not going to go into details at this point because I'm running out of time. Okay, when you get your own phylogeny, this is actually the point that it raises most contention in society when you're talking about evolution. Can we treat ourselves as any other animal? Yeah, nothing changed with us. We're just one more species of great ape. We are a species of African great ape that's evolved in those particular conditions, eventually became an invasive species and went all over the world, like pigeons, like cockroaches, like many other things. And as normal with invasive species, when they first enter in the environment, they create havoc. They just destroy everything, use all resources. Why? Because the normal parasites are not with them, because normal predators are not with them. All the difficulties that kept the population in check in the original area are no longer there. So they make a party. They just occupy everything, destroy everything. That's what we're doing right now. We're not unique in that. Many species do this kind of thing. We're just doing it in a grand way because we are so big. There are other species of hominids that occurred in many areas of the world. Of course, we're not the only one. And the time scale of all of them is more or less shown in this diagram here. Many people think, oh, okay, this means that this species is much more evolved than the other ones. No, it doesn't mean that it's more evolved. Okay, as I said, the time of evolution is the same for all terminals in a phylogenetic analysis. Of course, some of them were extinct, so you have to discount the part that they no longer exist. But the time of evolution is the same for all of them. Okay, just some splits are more recent than others. Just the split doesn't mean anything. There are species of worms which are separated by 10,000 years. Okay, that doesn't mean anything. It's in terms of how long you have evolved, just as a separate species. But the similarities between our own species and our closest relatives is really impressive. I mean, it's amazing that it took a while for people to realize that there is no difference, no fundamental difference, that we're basically the same thing, including cognitive abilities. Renato first called my attention to that chimps are a lot better than we are, even at some intellectual tasks, like remembering numbers on a screen. So this is
Scary.
scary. It's very scary. Yeah, it's humbling. Yeah. It is humbling. It's incredible that these things still exist, but they're not all killed. It's just because Congo is so big and the forest is so thick, our ancestors could not go in there and kill everybody. We're doing it now, but it is taking a while. Oh yeah, I forgot that. I still have to talk about parasites, otherwise Renato is going to kill me because he likes parasites so much. So I'm going to talk about this really quickly. This is a funny parasite. It is an isopod, similar to the group I showed you with the female in the belly of the male. It does a very interesting thing. When it's very small, it gets in the mouth of the fish, attaches to the tongue, and eats the tongue. But a host without a tongue doesn't survive too long. So and no parasite wants its host to die, because it will die along with it. So it has to do something. So what it does, it turns into an artificial tongue, a prosthetic tongue. It grows in the shape of a tongue and substitutes the tongue. It actually attaches to the bone where the old tongue was, and it serves as the tongue. I just found this was a cool example. Like this one. This is a crab. Most people say, people who know about crabs, this is a female crab because it's carrying this egg sac here. But no, this is a male crab actually. And these eggs are not the crabs. These eggs are parasites, which is actually a highly modified barnacle that transformed itself into this kind of cancer that goes inside the crab and changes its physiology and behavior. It makes the crab a species of robot for its own reproduction. It's a very impressive animal indeed. When it's free living, it's just like a normal larvae of a barnacle. But when it attaches to a crab, it turns into something very different, almost like a fungus that goes inside it and controls its nervous system so that it behaves differently. Like even male crabs behave like females. When the egg sac comes out, they go to the top of a rock and just shake the support of the egg sac to spread the eggs, just like a female crab does. The female crab does the same thing too. It's just that the male and female turn to the same behavior because of the parasites. But what is interesting in evolutionary terms about parasites is that they also have their phylogenies. And since they are attached to the host, usually their phylogenies tend to match the phylogeny or the pattern of ramification of the host. And we are part of this. We have examples of that on us. We don't need to go too far. For example, this is lice. Male-female lice. Human lice. We are like the dirtiest, in terms of lice, species of great ape. We have 3 species of lice. The other apes have 1. Well, anyway, I'm not too lucky. But this is 2 of them are actually subspecies. They're in the process of splitting, of being differentiated. So we have the head louse, we have the pubic louse, which is actually going extinct. But people shower too much these days. And the body louse, which is actually stays in the clothes. Its cycle is in the clothes. So its divergence from this, its sister species, is about the time when people start wearing clothes. Molecularly, it's about the same age as the oldest clothes known from humans, which is interesting. And this is the phylogeny of the lice. This is the phylogeny of their hosts. Notice that the phylogeny is similar. And this is what you find in parasite-host comparison of phylogenies. Usually their structure is very similar because they co-evolved. So the host splits, the parasite splits. So this is a very interesting way of seeing evolution in action again. If you don't find a structure of relationship completely unrelated to the structure of relationship of the hosts. They're usually related. Very similar beyond what would be statistically acceptable randomly. Much beyond, orders of magnitude. Okay, just to finish, since I talked about apes a little bit, many people think evolution is something new, and the evidence for evolution was only available after Darwin and things like that. I want to bring you back 250 years, or more than that actually, to Linnaeus, which is the founder of the present system of nomenclature that we use today. Just nomenclature, I mean, this thing is really old. Linnaeus published this book called Systema Naturae. He was a very religious man, dedicated to Jehovah, as everybody at that time. And in this book, he tried to classify the universe. The universe. It was very ambitious. He didn't want to classify just us or living things. He wanted to classify everything. So he starts with God. And he gives a definition. So if you meet God in the streets, you can tell, oh, hi God, it's you. Check all the characters here. World, astros, stars and planets and things, elements, earth, and so on. So he starts really broad and goes on to species of beetles, all the way. Eventually, he gets to mammals. No, the name is the same, mammalia. Blah, 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 blah. And within mammals, primates. Hey, Homo. We are there. Within primates, within mammals, within animals, blah, 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 blah. This guy was from the 18th century. And eventually he gets to other species of Homo. Notice the name here, troglodytes. The chimp is called Pantroglodytes today. Actually, this troglodytes is not the chimp. It's probably another great ape, the gibbon, probably. But Linnaeus, who was not an evolutionist, he placed us along with the other apes in the same genus, in the same order, in the same family, in the same everything. He noticed that. He noticed that the similarities were too much to be non-significant. It was impossible to exclude us from the rest of the living world. And that for a creationist living in the mid-18th century. Many people didn't like that. And this is a fabulous letter he wrote to humanity. This is not published, this is a letter. I mean, it's published as a letter. He didn't publish interesting things like that. He just wrote in letters. Look what he says. It does not please you that I have placed men among the anthropomorpha, that means primates. Perhaps because of the term with human form. But man learns to know by himself. Let's not quibble over words. It will be the same to me whatever the name we apply. But I seek from you and from the whole world a generic difference between man and simian that follows from the principles of natural history. I absolutely know of none. If I had called man a simian, or vice versa, I would have brought together all the theologians against me. Perhaps I ought to have done so in respect of the laws of our discipline. Well, with that, I will end my presentation. Thank you very much.